Okay, so section 23.2 is now talking about Hardy Weinberg. So we established in 23.1 that there's genetic variation within a population, and so we're just going to take that a little bit further. So basically, Hardy Weinberg is a set of rules, a principle, a mathematical principle that will allow us to determine whether or not a population is evolving. So if we're seeing changes in a population from generation to generation, we can use Hardy Weinberg to determine if the population is actually evolving, if those changes are due to evolution or to something else. So let's clarify what is a population. We did this a little bit last section, but basically a population is a group of individuals of the same species that live together and breed together. So all of the ants in room 100 are a population different from the population of ants in room 400, because the ants around here are breeding together, the ants over there are not, they are far away. And a gene pool is all of the alleles that are present in a population. So that is all of the alleles at every single locus. So every single allele that's present within a group of interbreeding organisms of the same species that live together. And we can talk about population and call it fixed at a specific gene, a specific locus, if there's only one allele present. So that means that if there's only the dominant allele present, then every single organism is going to be homozygous dominant. Or if there's only the recessive allele present, then every single organism is going to be homozygous recessive. So we call that fixed, if it's stuck there. So here are two different populations of caribou that live in the Yukon, up by the Arctic in Alaska. And so here's the porcupine herd and here's the 40-mile herd. They are the same species, and you can see that their geographies actually do overlap a little bit, but they tend not to mate with each other. For whatever reason, they remain two distinct populations for the most part. So they're not switching alleles or exchanging alleles by mating together in the next generation. So we can calculate the frequency of an allele in a population. So remember that if organism is diploid, which are all the animals that we're going to be talking about, they have two copies of each allele. So if there's 1,000 individuals, then there's going to be 2,000 alleles in that population, because they're all diploid, they all have two copies of the allele. So then we can also remember that if we can calculate the number of dominant and recessive alleles by looking at the genotypes of them. So if you are homozygous dominant, you have two copies of the dominant allele, but then the heterozygous individuals also have a copy of the dominant allele. So the number of dominant alleles is going to be the number of homozygous individuals times 2 plus the number of heterozygous individuals. Same thing with recessive. The number of recessive alleles is going to be the number of homozygous recessive individuals plus the number, or times 2, plus the number of heterozygous individuals. Let's look at that quickly. So if there's two alleles at a locus, which are all the ones we're going to be talking about, then we use P and Q to represent their frequencies. So P is going to be the frequency of the dominant allele, Q is going to be the frequency of the recessive allele, and so these must add up to 1 or 100% because there's only two possibilities. So the possibility of getting a dominant allele plus the possibility of getting a recessive allele together equals 100% because there's only two possibilities. Okay, so let's look at this population of wildflowers. It's incompletely dominant, so the heterozygous individuals are pink. The dominant are red, the recessive are white. So there's 320 homozygous red individuals, 160 heterozygous individuals, and 20 homozygous white individuals. So if we want to calculate the number of each allele, there's still only two. There's a red allele and a white allele. We look at the number of, so here's how we calculate the frequency of the red allele. We look at the number of homozygous red individuals, it's 320. These guys each have two copies of the red allele, so we do 320 times 2. And then there's 160 individuals that have one red allele, so plus 160 gives us 800. So 800 red alleles in this population of 500 individuals. And then, same thing for the white. There are 20 homozygous white individuals, and these guys each have two copies of the white allele, so we multiply that by 2, 20 times 2. And then there's 160 that are heterozygous, so 20 times 2 plus 160 gives us 200 white alleles in a population of 200 or 500 individuals. So then to find the frequency, our P and our Q of each allele, all we do is divide the total number of, or the number of alleles by the total number of alleles. So the P is the frequency of the red allele. So it's 800 divided by 1,000, because there's a total of 1,000 alleles, because there's 500 individuals and they each have 2 alleles. Gives us a likelihood or probability, a frequency of 0.8. So 80% of the alleles in a population are red. Same thing with the white. The Q is the frequency of the white allele. There's 200 white alleles out of 1,000 possible alleles, so the frequency is 0.2. And remember that these two numbers are always going to add up to 1 because they're the only two possibilities, so they always have to add up to 100%. So let's get into Hardy Weinberg now. So Hardy Weinberg is a principle that describes a population that's not evolving. So if a population is in true Hardy Weinberg equilibrium, it's not evolving. And we can predict the number of individuals of each phenotype in each generation. Okay, so the Hardy Weinberg principle states that frequencies of alleles and genotypes in a population remain constant from generation to generation. So in a situation where there's random mating happening, the likelihood of the allele frequency staying the same is 100%. So the number of P's and the number of Q's is going to stay the same from generation to generation. So, okay, looking at our population of red and white flowers again. So basically, oh, where did that one slide go? No. Here we are. Okay, so this is basically what happens in the real world. So the frequency of the red allele is 0.8, the frequency of the white allele is 0.2. So we just have to make sure that 80% of the alleles in this bucket are red and 20% are white. And so each member of the next generation is basically going to be grabbing 2 of those out at random. So the likelihood that you're going to get a red allele when you grab in there is 80%, and the likelihood that you're going to get a white allele is 20%. So let's look at this. So we can calculate the frequency of each genotype. So if we're going to talk about the frequency of being homozygous red, that means I reach in with my right hand, I grab a red. I reach in with my left hand, I grab a red. So with my right hand, the probability of getting a red one is 80%. My left hand, the probability of getting a red one is 80%. 80 times 80%, or 0.8 times 0.8, gives me 0.64. So the likelihood of becoming homozygous dominant, homozygous red, is 0.64 or 64%. Same thing with the white individuals. The likelihood of me grabbing in with my right hand and grabbing a white allele is 0.2. Same thing with my left hand, 0.2. 0.2 times 0.2 gives me 0.04. So the likelihood of being homozygous white is 4%. Now, the heterozygous individuals is where it gets a little interesting, because the likelihood of me grabbing in with my right hand and grabbing a red is 0.8. Grabbing with my left hand and grabbing a white is 0.2. 0.8 times 0.2 is 0.16. However, it could be reversed. I could grab a white one with my right hand and a red one with my left hand. That's also 0.6. So you have to add together those two probabilities. So it's 2 times 0.8 times 0.6 gives us 32% of the likelihood of being heterozygous. And you can also do Punnett squares with this, which is kind of crazy. So, okay, so here's my Punnett square. So 80% chance of getting a red allele, 20% chance of getting a white allele. Same thing on this side, multiply them together. So if I say, what's the likelihood of being pink? 16% this way, or 16% this way. So you have to account for both of those. So it's always 2 times P times Q. Okay. So if P and Q represent the relative frequencies of the only 2 possible alleles, we only have 2 alleles in this situation, then we can always say that the number of homozygous dominant plus the number of heterozygous plus the number of homozygous recessive is going to equal up to 1, because those are only 3 possibilities. So P squared is the frequency of homozygous dominant, 2PQ is the frequency of heterozygous, Q squared is the frequency of homozygous recessive, so they always add up to 1. So we said that there's very specific conditions for a population to be in Hardy Weinberg equilibrium. There's 5 of them. Remember, this describes a population that is not evolving. So the allele frequencies are not changing. In real populations, this doesn't really happen. So Hardy Weinberg is kind of a theoretical thing. It doesn't happen in real life. So the 5 conditions. This is on page 475 in your textbook. No mutations, because mutations add new alleles into a population, and we don't want that. Remember, we want our allelic frequencies to stay the same. Random mating. That means when I reach my hand into that jar of alleles, I can't look ahead of time, because if I do, then, or if I'm saying, oh, I just want red, or I just want white, then it's going to mess up the frequencies. But if it's completely random, then it will maintain the same allelic frequencies. No natural selection, so that means that all phenotypes and all genotypes must be equally fit and likely to survive and pass on their genes. There must be an extremely large population size, because that will remove any possibility of changes due to chance. And lastly, there must be no gene flow. So that means that we can't bring in new alleles or get rid of alleles, because then we're going to mess up our allelic frequencies. So those are the 5 conditions for Hardy Weinberg. So sometimes, remember, this is for when we look at Hardy Weinberg, we're talking about one gene, one locus. So sometimes populations can be evolving at a specific locus, like maybe this flower population, the allelic frequencies for the flower color are changing, but the allelic frequencies for plant height or seed shape are staying the same. So each allele evolves independently of other alleles. So then your textbook talks about how we can use this in real life, and basically we can use it to predict if evolution is happening in a population. So we can also measure or predict the amount of individuals carrying a specific allele for an inherited disease. So I have no idea how to say that word. Phenylketonuria, PKU, is a disease where you can't have phenylalanine. Basically, there's an amino acid that you can't eat, and so we can use this to predict the amount of PKU individuals in a population. You can read this in your textbook. And that is the end of section